Good morning to all of you. Before going to start my presentation, I would like to give the brief introduction of my institute and my teamwork. This is where it is. The name of my institute is State Forest Research Institute. It is situated in the central part of India. It was established in 1963. And he is me. He is my boss. He is my director. His name is Dr. G. Krishnamurthy. He is a very senior forest officer of Indian Forest Services. He is my Mr. Amit Pandey. He is a senior research officer of my group. These are my PhD students doing the PhD under my guidance. So this is brief about the introduction about my institute. The title of my presentation is to Standardization of Clonal Propagation Protocol of Dillenia pentagyna, an important and endangered tree species through stem branch cutting. You know that medicinal plants are very useful for humankind, and they are used since the time of civilizations. India is the 12th mega biodiversity center in the world, with the presence of 45,000 species of various species, including medicinal and aromatic plants. The traditional system of medicines is widely practiced at the global level at now, and many of them medicinal plants are going to be reared, endangered, and threatened due to heavy biotic impact on them. This is the taxonomical classification of the species which I have worked on them. The genus is Dillenia, the species is pentagyna. And it's have 100 species of this genus, Dillenia pentagyna. One of the interesting things that the species is named after the great German botanist, John Jacob Dellens. And in English, it is known as dog teeth. And in Hindi, in the local language of India, Hindi, it is known as karmal. This is the situation of its occurrence at the global level. It is mainly found in the tropical and subtropical parts of the world, like as Bhutan, India, Indonesia, Malaysia, Myanmar, Nepal, Thailand, Vietnam, and even in Australia, basically tropical and subtropical regions of the world. In India, it is mainly found in the central part, in the western part, eastern, the northern part, and the southern part of the country. It is found rarely in the forest areas due to overexploitation of this species. This is a long, huge tree attaining the height of up to the 15 meter tall. The trunk bark is basically yellowish, this grayish white in color here. The leaves are very simple, alternate, arranged, clustered on the plant. The flowers appear during the month of May and June. They are yellow in color. The fruit and seeds globulous, and as I already told that it is an evergreen found and semi-evergreen forest up to 1400 meter height. So this is detailed about the taxonomical detail of the plant species which I have worked. So many discussions have been discussed about the use of alkaloids today and day before yesterday. Regarding the importance of alkaloids and curcumin. So this also contains some of the important alkaloids, butylene, butylenic acid, deltylene, and so many flavonoids, terpenoids, phenolics, and saponins, which are used to cure different ailments. These are the medicinal use of the tree to use the curing of diabetes. It is available in the literature of the species. We have not conducted any kind of research or to use these. This is available through the literature. They are used to cure the diabetes, diarrhea, dysentery, pneumonia, and for hair growth also. Specifically this bark. In case of leaves, because this is an important plant, so many part is used for the treatment of different diseases. This is about the bark. This is about the leaves. It is used to cure the cut and wounds. Even it is used for the bone fractures, the bleeding, piles, and so many skin diseases, body pains, anti-cancerous, and so on. The Ayurveda system of medicine is very important in India. It is very old and ancient treatment system of medicinal use since long back, about 5,000 years ago. This system was developed by our Indian ancient people. They also recorded that it is used in such kind of diseases like fistula, wounds, diabetes, and so many diseases. It is already available in the Ayurvedic system of medicine. Okay, friends. We know that plant can be plant can be propagated by two methods. I have written a word here. This is recalcitrant. I think you can define what is recalcitrant. Yeah, the plant species which is difficult to propagate by conventional propagation methods like seeds. So it is highly recalcitrant tree species in nature. So why we have taken this species? You see, through seed, the average is about 40,000 seeds per kg. It's very small seeds. Germination is after 120 days. Seed longevity is very short, maybe of 1 month. The seed germination percentage is less than 1 to 2%. It's very, very poor to germinate by seeds. And if we go for natural plantation in the forest areas, we have to raise the plants by sowing the seeds directly into the field. This is the method of natural way of propagation of this species. Now, the second option we have adopted is clonal propagation. As my earlier presenter has already given the brief about micropropagation and tissue culture. And another method is macropropagation through stem branch cuttings. These are the methods of clonal propagation. Of course, there are several methods of clonal propagation, maybe grafting, budding, air so many. Okay. So, at present, there is no report at all reported across at world level regarding the propagation of this species by artificial means both macropropagation and micropropagation. Not reported well or documented so far. So this is the region we have selected the species for its study. Now, I'm again focusing about the objectives and need of the study. First of all, this is an endangered species. It's rarely occurs in the natural forest of India. As I already told that, it is recalcitrant in nature. That means it is difficult to propagate by any methods. Natural way, you can say. It is a highly commercial important tree species. As you've shown that it is used for the treatment of several diseases. So it is commercially exploited. The clonal propagation techniques or the methods are not well documented or reported till now. Therefore, we have tried to standardize and appropriate commercial propagation techniques using the stem branch cutting of these species through clonal propagation. This is the method how we have developed the techniques, methodology for clonal propagation through stem branch cutting. We have selected the phenotypically superior plant, or you can say the morphologically superior tree species from the natural grown areas. Then collected the various size and thickness of the cuttings with 1 or 2 nodes in them. Then the cuttings were properly washed with bebastine. It is a broad spectrum antifungal agent to kill the microbial load of the surface of the cuttings. Then we have given the treatment of indole butyric acid and naphthol acetic acid. They are the root promoting hormones to induce the roots from the cuttings, ranging from 100 to 5000 ppm. These are the parts per million concentration we have prepared to with the treatment of the cuttings. For 20 minutes. After treatment, the cuttings were placed under mist chambers. We have mist chambers there. You know about the mist chambers. They are the area where we can propagate the plants artificially. At 35 plus minus 5 degree temperature with intermittent misting systems, means we provide 2 to 3 times misting by automatically system to provide the to maintain the moisture around the cuttings. Then the observations were recorded periodically after 1 week. After the induction of roots from the cuttings, I have also used a word here, stacklings. Do you know the word stacklings? What is a stacklings? It is a technical word. No. Stacklings is it's when the roots is induced from the cuttings. When the roots induced from the cuttings is known as stacklings. All right? When they induce the roots induced from the cuttings, it is also known as stacklings or the complete plant. It is known as stacklings. So I have used, this word is rarely used by the scientists in the forum. And we have shifted these plants into the polythene bags at 1:1:2 FYM, that is sand, soil and FYM. The ratio was 1:1:1. The plants were maintained in the shade net for further hardening process. So friends, this is about the methods which we have used to raise the clonal plants of this species. Okay. So this is the results which we have achieved by the study. Standardization of clonal propagation protocol. These were the types of the cutting which we have used. That was 10 to 15 centimeter long. The thickness were 8 to 9 mm thick, like this. Is the type of cuttings we have used. The hormonal treatment which we have standardized for this species is 500 ppm concentration of IBA. Of course, we have used NAA also. We have used 2 kinds of root promoting hormones, NAA and IBA. But in case of NAA, we have not found any kind of results for inducing the cuttings, roots from the cuttings. So this is the standard protocol which we have standardized for this species for its clonal propagation. That is 500 ppm concentration of IBA. The treatment duration, as I already spoken about, 20 minutes. The physical condition of the mist chamber was, the temperature was 35 plus minus 5 degree. The intermittent misting by 2 to 3 times in a day. The culture medium we have used, that is medium grade sand. Why we have used this sand? Because it's maintained the moisture around the cuttings, appropriate moistures around the cuttings, so avoid to degrade the cuttings. That's why we have used this medium grade sands with small gravels along with sands. The hardenings were achieved at these combinations. These are the cuttings which we have placed under the mist unit for induction of fruits on sand. We have used sand as a culture media. Okay. So this is the final result we have achieved by this study. The rooting response was achieved within 30 to 35 days, and the optimum concentration of IVA was 500, and we achieved about optimum rooting response of more than 60% in this species. Because it is highly recalcitrant species, it is very difficult to propagate by other methods, but we have standardized the technique for its clonal propagation, true to type production of plants, true to type from nature to its office space. So this is the induction of roots from the cuttings. Beautiful roots we have achieved from the cuttings. You can see here. You can discuss with me in meantime also, in between my presentation. Okay. At the end of, okay. So this is the plants were shifted to the polythene bags and maintained in the hardening chamber in this chill-net house. And the plant establishment rate was more than 78% plants were survived after hardening. So this is again the great achievement of this plant, because it is difficult to propagate by seeds. So we have successfully developed the techniques of its propagation and production of plants by this method. So thank you very much.